Hey, I'm Kyle Gheres, and this is Thursday Brain Bytes. So last week we were talking about the biochemical and molecular phenomena that lead to synaptic currents in neurons in the brain. So this week we're going to talk in a little bit more detail about how we measure those synaptic currents and how the proximity of your recording electrode to where those currents are being generated actually influences the signals you're measuring and how that can affect interpretations of neural activity and the visibility of certain cells within the cortical system. So using a model, we'll look at kind of three different systems as an example here. The first would be a cortical micro-ECOG sitting on the surface of the brain. We would then have a laminar multi-site electrode and then a single-site microelectrode implanted into the granular layer of cortex. So in this case, we can think of this as a somatosensory cortex or visual cortex as a model system. And so using the laminar electrode, where we could sample across the entire length of cortex, if a stimulus is delivered where the ions are flowing into the cell, you would see a negative change, a negative inflection of the LFPs. So this would be a negative current, you would see an increase. However, farther away from where that synaptic influx is occurring, you actually see flow away from the electrodes as the currents balance. And then there's a reversal following rapidly thereafter as synaptic activity continues. So this biphasic response actually is captured as this flow of current source density, which can be broken down into these sources and sinks of current. And so when we think of things as forming a dipole, it's due to the neural nature being highly polarized. So in the cortical system, we have pyramidal neurons which stretch along this entire axis of the brain, with the dendrites way up at the superficial surface and the cell bodies and axons way down at the dorsal layers of the brain. When a synaptic current occurs up in the dendritic field, again, you would have a negative inflection as ions flow into the dendrites across the synapse, while the field potentials near the cell body are actually positive as ions flow away from the cell and towards the dendrites. The inverse would be true for a propagating action potential that's reaching the cell body. As the ion channels open at the soma, you have the negative inflection near the soma, while there is a positive ionic current flowing away from the dendrites outside. However, there becomes an issue when your cells are no longer highly polarized like the dendrite, or like pyramidal neurons. And so in the case of something like a layer 4 stellate cell, where your axons and dendritic fields are overlapping, you can have synaptic currents flowing into the cell. However, you have to be extremely close to that active synapse to measure the changes in field potential. This again is wholly true if there are somatic action potentials, as the lack of a polar nature inhibits the formation of large dipoles that can be observed through neural activity. So that's your Thursday Brain Bite.